In this presentation, we will introduce a microfluidic-based islet perifusion system for simultaneous measurement of dynamic insulin secretion, calcium influx, and mitochondrial potential changes of pancreatic islets in response to insulin secretagogues. The perifusion system is composed of a 3-layer microfluidic device, syringe pumps, and a fraction collector. Insulin secretagogues-induced calcium influx and mitochondrial potentials are imaged using fluorescence microscope setup.
Hi, my name is Adeola from the laboratory of Dr. Oberholzer in the Department of Transplant Surgery at the University of Illinois at Chicago.
Hi, I'm Dongyoung also from the Dr. Oberholzer's lab.
Hi, I'm Tricia Harvat, also from Dr. Oberholzer's lab.
Today we will show you a procedure for simultaneous islet imaging using a linear glucose gradient.
We use this procedure in our laboratory to study islet physiology and function.
So let's get started.
The microfluidic device used for this procedure is assembled from 3 layers of cured PDMS, generated by standard photolithography. The first layer, generated from a silicone master, has the wells of the microfluidic device that will gently hold the islets in place, 150 micrometers deep and 500 micrometers in diameter. The second layer has channels 500 micrometers tall and 2 millimeters wide. The middle of the channel is expanded to spread out the flow for better exchange of solution inside the device. An exchange well is made by punching a hole 3 millimeters high and 7 millimeters wide in the middle of the layer. The third layer is a thick slab of PDMS. To prepare this layer, punch small holes on both sides that align with the inlet and outlet of the channel on the second layer. Once all 3